Introduction. Welcome, medical students, to today's lecture on the intricate world of biochemistry. In this detailed script, we will explore the fascinating topic of glycogen metabolism and its crucial role in energy production, regulation of blood glucose levels, and various metabolic disorders. So, let us embark on this biochemical journey and dive into the depths of glycogen metabolism. To understand glycogen metabolism, it is imperative first to comprehend the biochemical pathways of glucose metabolism. Glycolysis, gluconeogenesis, and the TCA-Krebs cycle play instrumental roles in the conversion of glucose into usable energy within our cells. Glycolysis involves the breakdown of glucose into pyruvate, generating ATP and NAD as energy carriers. Gluconeogenesis, on the other hand, allows for the synthesis of glucose from non-carbohydrate precursors, such as lactate, amino acids, and glycerol. The TCA cycle completes the oxidation of glucose, providing reducing equivalents and energy-rich compounds, such as NAD and FOD2. Maintaining appropriate blood glucose levels is crucial for cellular homeostasis. Insulin and glucagon, hormones secreted by the pancreas, synergistically regulate these levels. Insulin promotes glucose uptake by cells, inhibits glycogenolysis, and stimulates glycogenesis. Conversely, glucagon stimulates glycogenolysis and gluconeogenesis, ensuring a constant supply of glucose during fasting or exercise. The balance between insulin and glucagon governs blood glucose levels, enabling a finely tuned response to physiological demands. Glycogenesis is the process by which excess glucose is converted into glycogen to be stored in the liver and muscles. Several enzymes and pathways are involved. First, glucose 6-phosphate is converted to glucose 1-phosphate by phosphoglucomutase. Then, glucose 1-phosphate reacts with uridine triphosphate, UTP, to form UDP glucose, catalyzed by UDP glucose pyrophosphorylase. Glycogen synthase elongates glycogen chains by adding glucose residues using UDP glucose as the substrate. The process continues until a branch structure is formed. Key players in glycogenesis are glycogen synthase and branching enzyme. Glycogen synthase, regulated by both allosteric and hormonal factors, facilitates the elongation of glycogen chains. Phosphorylation inactivates glycogen synthase, while dephosphorylation activates it. Branching enzyme, also known as amylo alpha 1,4,1,6 transglycosylase, introduces alpha 1,6 glycosidic linkages, creating branches in the glycogen structure. These branches enhance the accessibility of glycogen for enzymatic degradation. Glycogenolysis, the breakdown of glycogen to glucose, is primarily regulated by the enzyme glycogen phosphorylase. This enzyme catalyzes the release of glucose 1-phosphate from the non-reducing end of glycogen chains. Glycogen phosphorylase exists in two isoforms: muscle, glycogen phosphorylase B, and liver, glycogen phosphorylase A. Allosteric regulation, hormonal control, and covalent modification, such as phosphorylation, modulate the activity of glycogen phosphorylase, ensuring a balanced supply of glucose based on metabolic demands. Glucagon and epinephrine play vital roles in activating glycogenolysis during times of increased energy demand. Glucagon, released by the pancreas, promotes glycogenolysis in the liver through the CAMP-dependent pathway. Epinephrine, released by the adrenal glands, stimulates glycogenolysis in both the liver and muscles by activating adenylate cyclase and CAMP production. These hormones trigger a cascade of events that amplify the glycogenolytic response, ensuring an adequate supply of glucose for energy production. Glycogen storage diseases are a group of inherited metabolic disorders characterized by enzyme deficiencies within the glycogen metabolism pathway. These conditions profoundly affect carbohydrate metabolism. For instance, glycogen storage disease type I, GSDI, also known as von Gurke's disease, results from a deficiency in glucose 6-phosphatase. This leads to impaired glycogenolysis and gluconeogenesis, causing severe fasting hypoglycemia. Understanding these diseases helps shed light on the intricacies of glycogen metabolism's impact on overall metabolic health. Carbohydrate metabolism intimately interacts with lipid and protein metabolism, ensuring balanced energy utilization. Excess glucose can be converted into fatty acids and stored as triglycerides in adipose tissue, serving as an energy reserve. Furthermore, intermediates from glucose metabolism participate in various anabolic pathways, supporting protein synthesis and other cellular processes. The intricate interplay between these metabolic pathways is essential for maintaining overall metabolic homeostasis. Glycogen serves as an essential energy source during prolonged fasting or exercise when glucose availability decreases. Glycogenolysis provides a rapid supply of glucose to sustain energy production, enabling the body to meet its energy demands. Skeletal muscles predominantly utilize their glycogen stores during exercise, while the liver contributes to maintaining blood glucose levels. Understanding the role of glycogen metabolism in energy production aids in optimizing athletic performance and managing metabolic disorders. Glycogen metabolism plays a crucial role in the brain's energy supply, particularly in supporting neuronal and glial functions. Astrocytes store and release glycogen, providing glucose as an energy substrate for neurons, especially during increased cerebral metabolic demands. The dynamic regulation of brain glycogen levels influences various physiological processes and neuronal activity. Exploring the neurometabolic implications of glycogen metabolism enhances our comprehension of brain function and neurodegenerative disorders. The intricate interrelationships between glycogen storage and hypoglycemia are evident in different physiological conditions. Fasting, exercise, and certain metabolic disorders can lead to hypoglycemia due to increased glycogen depletion. Glycogen storage disorders, as mentioned earlier, can also result in fasting hypoglycemia due to impaired glycogenolysis. Analyzing these interrelationships allows us to recognize the complexities underlying hypoglycemic states and devise appropriate management strategies. Disruptions in glycogen metabolism can contribute to the pathogenesis of metabolic disorders like diabetes mellitus. In type 2 diabetes, impaired insulin signaling adversely affects glycogen synthesis and breakdown, leading to altered glycogen storage. Insufficient glycogenolysis also contributes to the elevated glucose levels observed in diabetes. Understanding the links between glycogen metabolism and metabolic disorders aids in developing targeted therapies and management approaches, offering hope for improved patient outcomes. Congratulations, medical students, on completing this in-depth exploration of glycogen metabolism. We have delved into various topics, ranging from the biochemical pathways of glucose metabolism to the links between glycogen metabolism and metabolic disorders. Glycogen metabolism's intricacies are crucial in providing energy, maintaining blood glucose levels, and supporting various physiological functions. Armed with this knowledge, you are now better equipped to comprehend the intricate world of biochemistry and its implications in medical practice. Now, step forward with confidence and apply this understanding to further research and patient care. Remember, the wonders of biochemistry await your future discoveries.